Hi, everyone. So I'm Ansia van Coller from the South African Medical Research Council's genomics platform, and I'm excited to tell you guys some more about the work we've been doing on hyenas from the Kruger National Park. But first, I would just like to use this opportunity to say something about the genomics platform, because I'm sure that several of you might not be familiar with this resource. So the SAMRC genomics platform was established in 2019, and it is a purpose-built sequencing facility, one of the first in Africa, and you can find more information on our website. We also have links to the first whole genome human sequence that was sequenced in South Africa. We do a lot of our own independent and collaborative research, but we also have a sequencing arm, sequence service arm. So we offer sequencing as a service. And the MRC is not a for-profit organization. The purpose of the platform is just to make sequencing more affordable and accessible to help boost genomic research in Africa. So for a bit of background on hyenas, so the spotted hyena is one of the largest predators that is native to southern Africa. They live in really large societies, far larger than any other mammalian carnivore. They are highly social animals, and they have very unique behavioral and ecological functions. They are known for their matriarchal system, where the females are much larger and more aggressive than the males. And you see this even in the very young cubs. They are what's known as hypercarnivorous, which means they consume not only the meat of their prey, but they are also adapted to consume the bones and every last piece. They are highly adaptable predators, so they can actively hunt for prey, but they also scavenge kills from other predators. So they are not opposed to eating leftovers, even if these leftovers have been left out in the sun for multiple days. And I suppose because of this scavenger lifestyle, they have evolved incredible resilience and resistance to infections, which makes them really interesting, to me at least. So the Kruger National Park is home to a very large population of spotted hyenas. I think the last estimate was up to about 10,000. So if you're going on a game drive, or you have been, you've probably seen a spotted hyena. Most animals at the Kruger are monitored, and the hyenas are no exception. So some of these animals have GPS collars, and there are camera traps everywhere, which researchers utilize to study their behavior, their movement, and their interactions with the environment, especially in those regions that are in close proximity to humans. They've also recently started looking at the feces and the blood of the hyenas to monitor their well-being and to check for infections. So it is important for them to screen for infections in animals within the Kruger to prevent the spread. And interestingly, these hyenas are less likely to have infections despite being in close contact with humans or other animals that carry a multitude of pathogens. So the spotted hyena is therefore a very fascinating model species that we can study for complex social behavior, evolution, sexual dimorphism, as well as their unique immunological and metabolic adaptations. And to decipher the genetic basis of these remarkable traits they have, this would be greatly facilitated by the use of a reference genome. So there have been previous publications on draft reference genomes for the spotted hyena, but these were derived from captive individuals, which can limit our insight into natural genetic variation. So the first publication was in 2020, where they sequenced a male individual of unknown age from the Frozen Zoo in San Diego in the US. And then 2 years later, there was a long-read sequencing done on a male specimen from the Beijing Zoo. This was a much improved assembly, but it was still incomplete, and again, from a captive individual. So we don't know how long these individuals have been away from Africa and how relevant they are potentially for research in Africa. So we were able to get DNA from 11 free-roaming spotted hyenas at the Kruger National Park. And here's some information on them. You can see different sexes, age groups, and all sorts of different regions within the park. We did long-read ONT sequencing on one of these individuals. We picked this one mostly because of the good quality and high concentration of DNA, which is a requirement for long-read sequencing, and the rest we did short-read whole genome sequencing for. So to generate the reference genome, I used the long-read ONT data, and this was a pretty basic pipeline. Anyone who's assembled a reference genome using long-read sequencing will be familiar with Fly, the de novo assembler, and I used Raycon to polish the assembly, and then Quast and Busco to assess the quality metrics of the assembly. And then this table just shows the quality metrics of our assembly compared to those published previously. And we can see that our assembly is a lot more contiguous and complete. So we have much longer fragments, if we go on the N50 and so, and fewer gaps. So the Busco percentage also showed that our assembly is a lot more complete, at 98.5% of the mammalian genes versus the 95% of the previous published ones. And so because we had sequenced 11 individuals, I thought it would be a fun idea to try my hand at making a pangenome. So this is a very simplified illustration of the pipeline that I used, but yeah, please do not consider this to be a tutorial. There are many different pipelines that can be used, and they are all applicable to different types of data that you have available. So how I went about this was to take the short-read sequencing data, which I aligned to our reference. I then did variant calling to find out, obviously, where each individual differs from the reference that we had built. I also extracted all the unaligned pieces of these individuals and did de novo assembly on those so that no piece of each individual's genome gets left behind. I used the variant call data and the reference to generate a consensus reference sequence for each individual, of course, including the little extra bits. I then relabeled all of the contexts so that I would be able to identify which individual they were from, because I then merged all of the genomes into a single FASTA file, which is the input for the progressive genome graph builder tool. Again, the disclaimer, there are many different tools. This was just the first one that I had the opportunity to test. So this is a very computationally intensive process, but eventually we were able to create a pangenome. So this pangenome was about 2.47 gigabases, which is a bit bigger than the 2.38 gigabases of the reference alone. But of course, the pangenome will incorporate these extra sequences that were not present in that reference individual. So it's a very complicated graph with about 35 million nodes and 50 million edges. So it does need some refinement still. And while I would love to show you a beautiful pangenome graph, I'm sure you can imagine that it is incredibly difficult to visualize such a complex graph structure in a 1D or 2D format. But I tried. So this is a one-dimensional rendering of the pangenome that was built, and it is highly compressed. So this red bit in the corner shows regions that have quite low coverage, so probably regions that are not shared between all individuals, and whereas the blue bits are high coverage regions, which are all individuals have. The black lines indicate links between the paths. So we have a lot of those. So this indicates topological differences in the graph, like twists and bends and things, which is an indication of variation, such as deletions or inversions. So Also keep in mind that this is not a chromosome level assembly that we had used, and that's why the chromosomes aren't necessarily grouped in order, which is why it looks like it's all over the place. But nonetheless, this is still a very useful reference that can be utilized. I will work on refining it a little bit, and we are considering generating a chromosome level assembly first and then to use that. Out of interest, though, I did try to construct a pseudo-chromosome for better visualization. So I did this by pulling out all the regions of the genomes that aligned to a chromosome from one of the closest living relatives, which do have a chromosome level assembly. In this case, it was the fossa from Madagascar. And you can see it's a bit easier to visualize some of the changes in the genome. But again, this is very compressed. It doesn't show all the details. So yeah, future efforts will focus on further refinement of this pangenome and potentially first generating a chromosome level assembly. And we're also working on annotating these hyena genomes, and we specifically hope to compare those highly conserved and the hypervariable regions to see if we can find genes that are associated with their resilience and their environmental adaptations. But yeah, overall, this study does present a significant advancement in the genomic resources that are available for spotted hyenas, with the first wild-derived reference genome and a draft pangenome, which contributes to conservation genetics, behavioral ecology, and comparative genomics. I'd just like to acknowledge the team at the SAMRC and at Stellenbosch University, who have all played very important roles in this project. And thanks, of course, to the ISCB organizers and to all of you for listening to my ramblings.
Thank you very much, Ansia. Are there any questions?
I'm not a hyena expert, so
Thank you very much for your talk. And I know many people think they are part of the ugly five, but I find them very cute, actually. So my question is, I have a couple of questions. First, did you explore the use of Hi-C to capture chromatin conformation and to have a better scaffolding, more contiguous? Second question, did you use flow cytometry? Do we have any real tangible idea of actually how many chromosomes are there? Because you have many more scaffolds than, I believe, chromosomes. And do you have an idea of ploidy for these individuals? Thank you.
Yes, so it is a diploid organism, to answer from the back. It is a diploid organism, and there have been studies, I can't recall now if it was flow cytometry based or like more karyotype based. So they do know that I believe it is 20 sets of chromosomes. We are hoping to do Hi-C to get the chromatin conformation, which we can use that information to do the scaffolding and to assign the chromosomes. We have done that successfully in our lab for other organisms. The difficulty that we have with the hyena is obviously we need to get fresh DNA from the exact same individual in order to do the Hi-C comparison. And they don't always manage to capture the same hyenas, or we'll have to confirm if we can get fresh sample for Hi-C sequencing, but I think that would be the best option. Thanks.
Okay, we've got another question here.
Hi, thanks for the talk. Do you plan to sequence hyenas living in other parks, or like brown hyenas or other types of hyenas, or is it just based in Kruger?
So we have only had access to the samples in the Kruger for this study, but we are not opposed to sequencing other species. We went with the spotted hyena because they're just far more common than, for instance, the brown hyena. But it would be interesting to see how those populations also compare to each other and also in different national parks.